Hey everyone, so I'm going to go over the microscope cell history and pond life investigation assignment. If you were absent that day, so you can fill out these slides. I mean, you might be able to do this one on your own just by looking up, but the next slide was me like demoing the microscope, and this is from an online reading. So if you can't see these, you need to zoom out a little bit. So I'm gonna go over, I'm not gonna keep my phone pointed at this, so just label it based off of what I go over on the microscope. So on the microscope, this part right here where you look through is called the eyepiece. This is a monocular microscope because it only has, like, you close one eye and look through. Some microscopes are fancy and have two lenses. So eyepiece, this that connects down to the lenses. This is called the body tube. Then you have the revolving nose piece because it rotates the lenses. So revolving nose piece. And there are 3 objective lenses. This one is low power and has a 4 right there. Oh, sorry, 4 right there. So this is low power, the smallest one. Then this one says 10. This is medium power. And that one says 40. That one is high power. This is the arm right here, or the microscope you have to hold it by when you're carrying it, and will be underneath the bottom part, which I'll go over. Okay, this platform right here, where you put your slide on, is called the stage. And this, where you put your slide, is called the stage clips. They might look different on the microscope. They might be like 2 little flat pieces that you put on top of your slide. But this one's kind of fancy because it holds it in place like that. Then underneath the stage right here, this little dial changes like the intensity of the light. So this is called a diaphragm right here. And then this is where the light comes out of. That is the light source. The bottom is called the base. We have 2 knobs. Sometimes the knobs are gonna be separate like this, one big, one small, and sometimes there'll be like a knob within a knob. So this one is the one that moves the stage
if we have anybody
a
that's
lot.
in 5th hour, Coach Williams' 5th hour weightlifting class,
See how much it moves?
please make your way to the
This is the
cafeteria.
course adjustment knob.
Anyone in Coach
This
Williams'
one,
5th hour weightlifting
like
class,
barely,
please make your way to the cafeteria. Thank you.
like you can't even see it move right now because it finally moves it. So this is called the fine adjustment knob, and that's the coarse adjustment knob. So that's the parts of the microscope. You'll use the microscope a lot more in zoology than you ever did in biology class. So it's important to know how to use the microscope properly in zoology versus biology. Then on this slide, I'm going to go over the answers. I'm not going to read the questions, but I'll do them in order and you'll write down the answers. So when you first, the very first thing you should do is turn your microscope on. But yeah, make sure it's turned on, the light will come out. So when you first start, you always want to start in low power. So that's the answer. Always start in low power and then work your way up as needed. Because you might be able to see everything in low power or medium power. You might not have to get up to high power. So always start in low power and then work your way up. So the correct total magnification of the lenses, even though like this one says 4x, is more than 4x. So the eyepiece has a 10x in it, 10x lens. So when we have 10x and 4x, the low power becomes 40. So you might want to put low power and then put like a colon, put 4 and then put an and then arrow. You always do dash dash and then the greater than sign, and it'll turn into an arrow, or however you want to write it. So the 4x will become 40. Medium power says 10. So if this is 10 and this is 10, the total magnification for medium power is 100. And under high power, since it says 40 and this is 10, the total magnification for high power is actually 400. You can get some fancy microscopes that have a fourth lens, that's a 100x lens, that can allow you to see bacteria, like actual bacteria a lot better. And if that's 100 and this is 10, that would actually be 1,000, but we don't have one of those microscopes. Yeah, like I was saying before, another reason why to start in low power and work your way up is because if I started off in medium power and it was never in focus, or I start in high power, it's gonna be very, very hard to focus whatever you're trying to find on your slide. So it's always best to start on low power to find your specimen. Structure you use when under low power to move the stage closer to the lens. So the first thing you want to do once you're under low power, in order to find whatever's on your slide, sometimes it might be all the way down, you want to use your coarse adjustment knob, see it stops, as close as possible to your lens. So you use a coarse adjustment knob to bring the stage as close to the low power lens as possible. Then when you're looking in through the microscope, if it's still blurry a little bit, then you'll slightly turn. You might have to play with it and turn it one way and then the other way to get it focused, but you're gonna slightly turn this if you need to get it into any more focus. And then once you get into focus, now you will only, that's another thing to put down on that one, only use a coarse adjustment knob under low power. You won't use this again, because once I move the medium power now, I'm only going to use a fine adjustment knob now to get it in the focus. Okay, once after I use this and now it's in focus, I can now go to high power. But look what happened. If I move that coarse adjustment knob, look how close it is to the lens. I could break the lens. I could break the lens on the actual objective lens. I could break the slide. So you don't want to use this again. You only use a fine adjustment knob. And that's why you have to start in low power and work your way up. Structure that can be adjusted to change the intensity of light from the light source. So actually sometimes it's actually easier to see specimens when we looked at the yeast. It's actually easier to see it when it was actually darker on the slide, honestly. So right here on the diaphragm, I don't know which way to turn it. You'll just have to usually just play with it to get the correct light intensity. So actually being a little bit darker actually allows me to see things a lot better. And then when the microscope is not in use. So when you are finished with the microscope, well, you need to take off your slide, because you might have a prepared slide. And the prepared slides are like the ones that are bought from like the laboratory companies that have like a specimen on there and the slide is labeled and it's sealed on there. That's a prepared slide. That would go back like in the box. But these would need to be washed and cleaned and put back on the paper towel back in the lab. But yeah, remove the slide, return it back to low power, and turn off your microscope. So don't leave that light running. You can leave it plugged in because other classes will be using it all day, but turn off the light so it doesn't burn out that bulb. So when you're done with it, that's what you do when making a wet mount slide. So like I said, prepared slide is the ones that already have like a laboratory label on there with specimen, it's sealed and it's bought from the company. So wet mount slide, I'm making it, I'll go back here. Like here is a pond water. So you have your slide and you have your cover slip. And I'm making wet mount slide, whatever liquid you have. Like when I worked as a vet tech, I had to look at fecal samples, urine samples, blood samples, ear swab samples, but anything that's liquid, well, with the pipette, at least with like this, you want to press the bulb first and then put in the liquid and suck up And then you're gonna place like 1 or 2 drops on your slide. So not too much, because if it's too much, it'll like spill out of the cover slip. So first thing you need to do when making wet mount slide is put a drop of the liquid sample on the slide. But I wouldn't put it on the microscope like this, because the lens is going to touch the water, and water is going to get all over it. Water is going to go everywhere. So you must put the cover slip on top of the sample. So put the drop of the liquid sample on, and then cover it with a cover slip. Then you can place it on the stage with the stage clips. This is a fancy microscope. Well, kind of fancy. Look at this. It moves the slide from left to right. I don't know of a name for these, but some of the cheaper microscopes don't have this. You can move the stage front to back, which moves the slide front and back. And then this one moves the stage clip and the slide left and right. So if you're trying to move around on the slide to find something, you would want to go all over the slide trying to find stuff. So you use these knobs. I'm trying to observe your specimen. So that's the microscope basics. The last thing you need to do is click on this reading, and you're going to write a summary about each of these scientists who are famously known for their discoveries with the microscope and the cell. So we have, back in the 1600s, we have Robert Hooke, I love these guys' names, Anton von Leeuwenhoek, Theodor Schwann, and Matthias Schleiden, and Rudolf Virchow. So from the reading, you're going to summarize like their discovery. Then you're going to write the 3 parts to the cell theory, which is on the page, is bullet pointed. And then in the Canvas assignment, there's a link for a video, and watch the wacky history of the cell theory, which completely goes over all of this right here. It goes over all these scientists and talks about their pettiness between each other too. So watch that video, and the link is in the Canvas assignment. So when you're done with this, when you open this, your autosave should be on. And as long as your autosave is on, when you go back to Canvas, and you're in the assignment, you can refresh your page, and your work should show up right on that Canvas page of this assignment. And then you hit the submit button, and it's tied. You don't have to save it, you don't have to attach it, it's already there. So all you have to do is hit submit, as long as your work is showing up there, like on the preview of the PowerPoint on Canvas. So if you have any questions, well, I'm not going to go over the pond water. It's hard to show all the different stuff that we found in the pond water. Like we found little nematode worms, we found insect larvae, we found some copepods, water fleas, all sorts of little protists and rotifers, little microscopic organisms. So we found a lot of cool things. I wish I had a microscope that could take pictures, which I could show the people who are at home, but I can't. It's hard to put my camera on the microscope to show what it's showing. But well, that's the microscope cell history and pond life investigation. There's nothing that the students had to write down or do for the pond water part. So just submit this and you're good. If you have any questions, let me know.